Welcome. Today, we'll be exploring the application of artificial intelligence in healthcare from a chemistry and engineering perspective. In recent decades, we have seen a dramatic increase in both structured and unstructured healthcare data, alongside a rapid evolution of machine learning algorithms. Artificial intelligence is positioned to play a key role in addressing current healthcare issues like availability, cost, and efficiency. It also provides novel approaches to previously unsolvable tasks through augmentation, automation, error reduction, prediction, and personalized precision medicine. However, the application of AI in healthcare is not without its challenges. The authors identify several key concerns, including the quality and availability of data, ethical, social, and privacy concerns, and issues with algorithm development and implementation. These implementation challenges encompass problems like a lack of explainability, data leakage, overfitting, and bias. To address some of these issues, the concept of explainable AI, or XAI, has emerged. XAI aims to tackle the black box problem inherent in some machine learning models, paving the way for a more trustworthy AI in future healthcare. Problems like data leakage, overfitting, and bias can be minimized or even eliminated with a periodically updated and carefully monitored data pipeline. As more practitioners join the effort, data quality and availability are expected to improve. Meanwhile, ethical, safety, and social concerns will likely rely on governmental or organizational regulations to build a robust framework. AI for healthcare can be applied across three main scenarios: basic biomedical research, translational research, and clinical practices. In basic biomedical research, AI can assist with tasks like gene function annotation, molecular dynamic simulation, and predicting transcription factor binding sites. For translational research, AI applications include biomarker identification, drug discovery, drug target prioritization, toxicity prediction, and genetic variant annotation. Finally, in clinical practices, AI is used for a wide range of tasks: disease diagnosis, risk stratification, medical vision, genome interpretation, treatment planning, patient monitoring, and robotic surgery, as well as managing population and chronic health.
The paper highlights a review by Zhang and colleagues that examine the motivations, data types, and algorithms used in healthcare AI. A central figure in the paper illustrates the entire workflow. It begins with the generation of clinical data, which includes electronic medical records, or EMRs, electrophysiological data, EP, images, and genetic information. This raw data then flows into two interconnected domains. Machine learning takes this data as input, while natural language processing takes clinical notes written in human language. These two processes enrich each other, generating more refined data for analysis. The ultimate output of this pipeline is the support of clinical activities, such as screening, diagnosis, and treatment. To give us a sense of what's being used in the field, the authors provide a pie chart showing the machine learning algorithms most commonly found in medical literature, based on a PubMed search. Support vector machines and neural networks are clearly the most popular, especially for imaging, genetic, and electrophysiological data. They are followed by logistic regression, discriminant analysis, random forests, and a variety of other methods.
A more recent review by Bohr and colleagues divides medical AI applications into 5 categories: medical vision for diagnosis and surgery, precision medicine, which is based on a patient's personal data like genetics and EMRs, intelligent health records, robotics and AI devices, and ambient assisted living. From the chemistry and engineering-oriented perspective of this paper, the focus will not be on health records or robotics. Instead, the authors expand the concept of precision medicine into 4 key topics: medical vision, diagnosis, protein design, and discovering small molecule drugs. Let's begin with the first of these topics, medical vision. AI in medical imaging encompasses various modalities like X-ray, MRI, CT, ultrasound, and pathology microscopy. These tools are used for the detection, diagnosis, and classification of diseases. While a comprehensive review of all publications is beyond the scope of this work, the authors highlight key aspects, including the advantages, challenges, and limitations of using AI in medical imaging. For instance, one figure presented shows the training procedure for a machine learning tool called EasyReg, which is used for multimodality brain MRI registration. Another figure illustrates the inference procedure for the same tool. AI offers several advantages that could revolutionize the medical imaging field. These include improvements in accuracy, speed, scalability, and cost-effectiveness. A key strength of AI, particularly deep learning, is its accuracy. These algorithms can discern patterns in medical images that are too intricate for even the expert human eye. Several studies have demonstrated the superior performance of AI in these applications. For example, a study by Nam and colleagues showed that an automatic detection algorithm outperformed physicians in classifying radiographs and detecting malignant pulmonary nodules on chest X-rays. Interestingly, the study also showed that when used as a complementary tool, the AI algorithm improved the physician's own detection performance. Other algorithms have been used for tasks like detecting lymph node metastases in breast cancer under tight time constraints. AI-based tools are already being developed and deployed. One such tool is the Automated Retinal Disease Assessment, or ARDA, which assists in detecting diabetic retinopathy and is being expanded to identify other diseases. AI and machine learning have also been applied to image analysis for various cell microscopy modalities, where they have overwhelmingly outperformed traditional non-ML-based tools. They excel in tasks like object detection, feature extraction, classification, and segmentation, particularly in radiology, pathology, and immunology. The proliferation of open-source software and new neural network architectures has significantly improved the accuracy of cell detection and segmentation in microscopy images. AI techniques have also improved the objectivity and sensitivity of cervical cancer screenings using whole-slide imaging. They've even introduced new methodologies like AI-powered transmitted light microscopy, which allows for the visualization and analysis of live cell structures without the need for labeling. Another notable advantage of AI is computational speed. Rapid analysis can lead to quicker diagnoses, potentially saving lives. An AI model called CheckSpert, developed by the Stanford Machine Learning Group, demonstrated this by analyzing a chest X-ray in just 10 seconds, a task that took a human radiologist over 20 minutes. Similarly, in cell microscopy, AI techniques facilitate rapid analysis, automating processes that would otherwise take humans hours or even days. This automation helps in extracting quantifiable cellular features, which can shed light on cell organization in various pathologies and streamline complex analyses.
Furthermore, AI systems are highly scalable, allowing them to manage vast amounts of data and assess multiple image sets simultaneously. The more data an AI is trained on, the more refined and accurate it becomes. This is especially beneficial for large-scale screenings or tracking disease progression over time. While large annotated datasets are scarce in medical imaging, recent progress in self-supervised models and those that leverage vision transformers and large language models promise the development of foundation models. These novel neural networks can learn from large, unannotated datasets and produce high-quality predictions. They eliminate the need for conventional supervised labeling, granting them the ability to perform zero-shot learning in diverse scenarios. A recent example is the Segment Anything model, which has demonstrated impressive zero-shot segmentation capabilities across various natural image databases. These models signify a shift from task-focused models to those that can adapt dynamically. If tuned for the medical sector, such models could significantly reduce the need for specialized single-task models. A single scalable foundation model could cater to multiple needs, leading to greater efficiency and quicker adaptability to evolving medical scenarios.
To provide a concrete example of how these systems are trained, the paper shows a diagram of the training procedure for EasyReg. The process starts with training data in a standardized space, known as MNI space. A mini batch of this data, consisting of synthetic images with random resolution and contrast, is fed into a convolutional neural network, or CNN. The generator creates the formed labels and auxiliary semantic labels. The CNN then predicts forward and backward fields. The process is guided by two loss functions, a similarity loss, which measures how well the prediction matches the target, and a regularizer loss, which helps prevent overfitting. The blue arrows in the diagram follow the layers of the neural network, while the green blocks represent the different terms of the loss function.
Of course, AI in medical imaging is not without its limitations. A significant challenge is the need for vast and diverse training datasets to ensure algorithmic robustness. Biases in training data can lead to misdiagnoses or reduced sensitivity in diverse patient populations. Another concern is the black box nature of some algorithms, where the decision-making process isn't transparent, making it difficult for clinicians to trust the AI's conclusions. There's also the challenge of seamlessly integrating AI into existing clinical workflows. Finally, regulatory and ethical considerations are pivotal in defining the boundaries of AI applications in this sensitive field.
The paper provides another helpful diagram illustrating the inference procedure for EasyReg, using an axial FLAIR scan and an isotropic MR-APG scan as inputs. The process begins with the input scans in their native space. Segmentation layers are applied to identify subcortical structures and cortical parcels. The centroids of these labels are then used to compute an affine transform to the standard MNI space. This transform brings the scans into the same voxel space where the CNN was trained. The CNN then computes the forward and backward deformation fields, which are combined with the affine transforms to finally deform the original images to match the reference.
Building on the foundation of medical imaging, the authors move to the broader topic of diagnosis. Diagnosis is a critical part of our healthcare system, and the advances in big data and AI can significantly improve the efficiency and accuracy of current diagnostic techniques. A comprehensive review of studies from 2012 to 2019 found that the performance of deep learning-based diagnostics is equivalent to that of healthcare professionals in classifying diseases from medical images. More recently, during the COVID-19 pandemic, AI-based diagnosis played an important role, achieving impressively high accuracy. A series of articles have elaborated on machine learning-based diagnosis for specific diseases like cancers and mental disorders, or using specific tests like electrocardiography and radiology.
A figure in the paper showcases several diseases and the machine learning techniques used to diagnose them. For heart disease, algorithms like BayesNet and support vector machine are listed. For diabetes, naive Bayes and various boosting algorithms are common. For liver disease, SVM, naive Bayes, and random forest are used. This highlights the variety of approaches being applied to different diagnostic challenges.
The fusion of extensive electronic health records, or EHRs, with machine learning holds great potential for advancing diagnostic capabilities. EHR data typically consists of patient demographics, medications, procedures, lab tests, and vital signs. By applying data-driven methodologies to these large datasets, researchers can investigate patterns and uncover true signals within the big data. Even before the machine learning era, data-driven approaches were used to detect safety signals in EHRs. Now, ML and AI algorithms can be incorporated to screen structured EHR data more effectively. Deep learning, in particular, has a profound influence on analyzing the heterogeneous characteristics of EHR data, from clinical notes to medical imaging. By transforming raw data through multiple layers of nonlinear modules, deep learning can amplify hidden features and improve EHR analytics. This can lead to timely disease detection, efficient diagnosis for high-risk groups, and precision medicine with tailored treatment plans. Patient health records can also originate from diverse data sources, including complex omics data like genomics. High-throughput technologies have produced vast amounts of this data from sources like the Cancer Genome Atlas and UniProt. Machine learning is well suited to manage and mine valuable information from this high-dimensional biomarker data. For example, Lewis and Kemp developed machine learning classifiers using metabolic biomarkers to investigate tumor radiation response, dividing tumors into radiation-sensitive and resistant types. Another study by Manak and colleagues used machine learning with dynamic live cell biomarkers to stratify cancer patients at high risk of adverse pathology before surgery. These examples demonstrate how machine learning can overcome limitations in traditional diagnosis and generate clinically relevant principles.
Next, the authors turn their focus from diagnosis to the molecular level, exploring the role of AI in the world of proteins. Proteins are complex macromolecules made of amino acid chains that are essential for countless biological functions, from catalyzing biochemical reactions to providing structural support. Understanding protein structure is critical in drug design, as proteins are often the target of inhibition. AI can automate and optimize the protein design process, enabling the development of proteins that are personalized, robust, and highly functional, sometimes unlike anything found in nature. However, challenges like imbalanced data and overfitting persist in this domain. Tools such as AlphaFold, AlphaFold2, and RosettaFold can accurately predict protein structure or assist in de novo protein design. Machine learning is also used to predict protein-protein interactions, protein function, and protein stability, as well as to advance enzyme engineering. As with any AI-assisted field, providing accurate training sets is essential. AI techniques with deep learning can analyze massive data sets of known protein structures to build models that predict how a new protein will fold based on its sequence. AlphaFold, for instance, has predicted the structures of more than 200 million proteins, covering almost every known protein on the planet. This AI network shortens the cycle of solving protein structures compared to conventional methods like X-ray crystallography, though it may have lower accuracy for less defined structures like intrinsically disordered proteins.
A diagram in the paper outlines the schematic workflow for constructing a machine learning predictor and its associated challenges. The first step is preparing the data. Data from experiments, computations, and databases are collected and turned into a table format. This data is then split into training and test sets. It's crucial to account for any errors, biases, or imbalances in the data at this stage, such as missing values or imbalanced datasets. The second step is training the predictor on the training set. A decision boundary is derived to classify future input. Here, the challenge is to balance between overfitting, where the model explains noise rather than fundamental dependencies, and underfitting, where it fails to account for complex dependencies. The final step is validating the predictor using the test set. Various metrics like precision, recall, and specificity are calculated to evaluate performance. Challenges in this step include skewed estimates from data imbalances and data contamination, where the test set is inadvertently included in the training process. While protein structure is crucial, enzyme kinetics, the study of reaction rates, is equally significant for protein engineering. Recent studies have deployed machine and deep learning methods to predict the turnover rate, or KCAT, of uncharacterized enzymes. This provides key insights into protein functionality. A figure illustrates this process of using machine learning to predict catalytic turnover numbers for genome-scale metabolic models. The process gathers features from diverse classes, including network properties, structure, biochemistry, and assay conditions. Machine learning models are then built to predict both the in vitro KCAT and the in vivo maximum apparent turnover rate, KAP-max. These inferred models are then used to parameterize genome-scale models. The performance of various machine learning models, from linear models to deep neural networks, is shown in a box plot, demonstrating their effectiveness. Another diagram details how experimentally measured KCAT values are downloaded from databases like Brenda and Uniprot. This enzyme information is then represented by numerical vectors using natural language processing models. The chemical reactions are represented using integer vectors. These concatenated representations are used to train a gradient boosting model to predict KCAT. The fitted model can then be used to parameterize metabolic networks, influencing predictions of proteome composition and growth rate. Although predicting enzyme kinetics with computational tools isn't new, these AI-aided approaches offer numerical fingerprints that consider the complete set of substrates and products. This allows for direct comparison of enzyme catalytic efficiency, adding a critical aspect to AI-based enzyme engineering. Moreover, these tools help us understand enzyme performance not just in vitro, but also their in vivo behavior. With proper safeguards, AI for protein design holds significant promise for revolutionizing the field, enabling more effective therapies, sustainable food production, and eco-friendly industrial processes. To collect the robust and reliable data needed for these models, a variety of high-throughput methods are used, as illustrated in another figure. Next-generation sequencing, or NGS, allows for high-throughput analysis of DNA and RNA. High-throughput screening methods, including robotics, fluorescence-activated cell sorting, FACS, and microfluidics, can screen millions or even billions of variants per day. Finally, deep mutational scanning combines high-throughput screening with NGS to comprehensively analyze sequence-function relationships in enzymes. From the world of large proteins, the discussion now shifts to the fourth and final topic in this section: small molecule drugs. The crystal and molecular structures of organic small molecule drugs have a profound impact on their bioavailability and medical applications. Machine learning has made impressive achievements in drug discovery, such as the discovery of the powerful antibiotic halicin. A flowchart in the paper shows a deep learning algorithm that uses graph neural networks and sequence-based models for small molecule drug design. This process often starts with a known molecule, represented by its SMILE string, which is then converted into a molecular graph structure. A generative model, combining a sequence-based model and a graph neural network, proposes new candidate molecules. A discriminator then evaluates these candidates, providing a reward that guides the generator to produce better molecules over time. With the onset of COVID-19, various screening methods were employed to discover novel inhibitor molecules for the main protease of the SARS-CoV-2 virus. These included using free energy perturbation calculations to design novel analogs and high-throughput virtual screening to identify competitive inhibitors. The utilization of AI in drug discovery for COVID-19 has gained considerable interest. Generative AI has been used to produce datasets of potential drug molecules, and researchers have used it to design small molecule inhibitors for various viral proteins. Looking forward, machine and deep learning-based AI algorithms have significant potential to accelerate drug discovery across the entire pipeline, from target identification to personalized therapy. Having explored these specific applications, the authors broadened the scope to discuss three related topics that underpin the use of AI in science and engineering. The first of these is the relationship between physics and AI. The author suggests that this relationship is complex. We need to be aware that AI is not only capable of generating data-driven insights from large datasets, but may also make profound discoveries in physics and chemistry that are difficult for humans to understand. A diagram of a multi-scale hierarchy of computational methods illustrates this point. Different methods operate at different time and length scales, from the exact quantum mechanics of individual atoms at the femtosecond and picometer scale to continuum methods that model material failure over hours and meters. Each method has its own limitations in accuracy and speed. Machine learning can benefit greatly by incorporating insights from these physics-based methods to improve interpretability, generalization, and efficient learning. An example of this physics-based machine learning is shown in a detailed figure on learning the assembly mechanism of ions in water. The method iterates between sampling transition paths of a molecular system and learning a committer, which is a function that predicts the probability of the system reaching a certain state. A neural network models this committer based on molecular features. At convergence, symbolic regression distills an interpretable expression that quantifies the molecular mechanism. This process was used to understand the formation of a Li-Cl ion pair in a simulation, and the learned model demonstrated both self-consistency and the ability to generalize to other systems like NaCl and NaI through transfer learning. The second related topic is model explainability. Although deep learning and neural nets are powerful tools, an explainable AI is crucial for trustworthiness, refinement, and fairness. As AI permeates critical infrastructure and decision-making, explainability becomes essential. Interpretable AI can work with people by providing meaningful justifications for its behavior, rather than just numerical confidence scores. The future lies in balancing sophisticated machine learning with human intuition, moving towards a future of human-AI collaboration. The third related topic is autonomous experiments. The paper has already shown examples in additive manufacturing and flow chemistry. AI and machine learning have significant potential to enable automated experiment design, adaptive sampling, and closed-loop control. One figure illustrates a machine vision control system for 3D printing, where a feedback pipeline enables online updates to extrusion parameters. A camera captures a snapshot of the nozzle region, a CNN predicts printing parameters, and the system analyzes a sequence of these predictions to determine if adjustments are needed. This allows the system to autonomously correct for issues like under or over extrusion. This integration of AI, machine learning, and robotics with a scientist in the loop can accelerate science and technology. The general concept of self-driving labs is becoming a reality, shifting the paradigm from a traditional make, measure, simulate approach to an integrated, self-driven approach for discovery and innovation. Finally, the authors address the crucial topic of security, ethics, and governance. To integrate physics-based explainable AI with autonomous experimentation, it's essential to have a mechanism to guarantee the underlying quality. This includes considerations of security, ethics, and governance, which often broaden into areas of infrastructure, social behaviors, and law. All parties involved need to take into account 5 ethical principles: transparency, fairness, doing no harm, responsibility, and privacy. A multidisciplinary and adaptable framework is required for effective governance, especially in sectors like government, education, and healthcare. For AI models themselves, it's well known that a biased training dataset can lead to poor quality output, garbage in, garbage out. A figure in the paper highlights the importance of the bias-variance trade-off. Supervised learning algorithms must balance 2 sources of error, bias and variance. A highly biased model is based on flawed assumptions and tends to underfit the data. Conversely, a high-variance model follows small variations in the data too closely, picking up random noise and overfitting. The optimal trade-off minimizes the generalization error of the model, how well it performs on unknown data. This can be estimated using cross-validation techniques, which involve splitting the data into training, validation, and test subsets to find the best model hyperparameters. In their outlook, the authors conclude that many fields will continue to be augmented by AI. In healthcare, AI will directly impact human intervention to prevent critical diseases and provide solutions for future pandemics. In manufacturing, AI can explore vast design spaces to discover new materials and devices.
AI both guides experiment design and predicts results to accelerate discoveries in batteries, sensors, electronics, and renewable energy.
To summarize the landscape, the paper presents a final diagram connecting notable champion models to their respective application niches. We see models like anomaly detection and classification being applied to additive manufacturing and battery technology. Bayesian optimization is linked to chemistry and flow and sensors. Neural nets and transformers are central to medical vision, diagnosis, pharma, and natural language processing. This visualization encapsulates the broad and revolutionary potential of data science, machine learning, and AI across chemistry, engineering, manufacturing, and health industries. The real promise, the authors conclude, lies in leveraging AI to connect advances across these fields, achieving exponentially more through collaboration than any one area might alone.